Welcome to the Science Lab. Today we'll be doing how do pigments allow plants to be producers. We will need the following equipment: a 500ml beaker, a hot plate, a test tube rack, 3 test tubes, methylated spirits, 3 250ml beakers, test tube holder, tongs, forceps, a spatula, masking tape, a pen, 3 different types of plant leaves. We will use silver beet, spinach, and red lettuce, 3 chromatography strips, and a beaker test tube adapter. You'll need to half fill the 500ml beaker with water and place it on the hot plate and bring to a gentle boil. This will be done for you already. You then need to label your beakers and test tubes with the different leaf types. You'll be given a small piece of each leaf type. Fold up one of the leaf pieces and using the forceps, carefully lower it into the water for 5 to 10 seconds to soften the leaves. This will make them easier to place into the test tube. You can also use the spatula to help push the piece of leaf to the bottom of the test tube. Now repeat this for each different type of leaf. You need to take the beaker test tube adapter and place it on top of the beaker. This is to hold the test tubes in the water bath safely. We now take the methylated spirits and fill to the 10ml line on each test tube. We then place the test tube into one of the test tube holes and into the water bath. You then repeat this step for the remaining test tubes. We leave the test tubes in the water bath for approximately 5 minutes or until the liquid has gone green and leaves have lost their colour. Once we have done this, we turn off the hot plate. Now using the tongs, gently lift the test tube up and then grab the test tube securely with the test tube holder. Do not use your fingers. Carefully pour the liquid into the matching labelled beaker, but don't pour the leaf in. Then place the test tube into the test tube rack to cool. We repeat this for the remaining test tubes. Leave the solution to cool for a minute or 2, then take your chromatography strips, make sure they're labelled, and place one end in the solution so about 3 to 4 millimeters of the strip is sitting in the solution. Fold the strip over the top of the beaker to hold in place. Repeat for all the strips and then leave for 10 minutes. We then take the strips out and place on paper towel to dry for a few minutes. You then need to measure each colour band and record this and other observations in your table. Pause here to avoid revealing the results. You now have all 3 strips. You can pause at each strip to take measurements and record observations in your table. Thank you for watching.